this time and this space with us for another MAPS talk. And I am incredibly pleased to introduce my colleague, Georgete Oraha, who will be speaking to you this evening. So Georgete graduated from UC Stanislaus with a Bachelor's of Science with a concentration in ecology in 2017. And then she went on to work on her master's. And during the course of her master's work, she did a research project which we are going to enjoy the results of and really sort of experience some of what she discovered, the highs, the lows, the drama, the doing science through COVID, all of the things that happened on her journey. And then she finished her master's degree at USC Fullerton.
CSU.
CSU,
Both CSUs.
I'm sorry. I knew it. CSU Fullerton in 2021. And she has recently come to us. So she's our colleague here in the biology department. So I just want to say, one reason I was so excited also to introduce her is Georgete is so passionate and so excited. She's an amazing educator because her enthusiasm comes through in all that she does. And she also has amazing taste in science-related wall decor in her office. You can tell things when you see how people decorate their space. And when I went and talked to her the first time, I was like, we both had women in science up on our wall, the same thing. So please join me in welcoming Georgete tonight for this talk. Thank you.
All right, everyone, can you guys hear me in the back? How are we doing? Yes, reassuring head nods, great, I love that. Okay, so thank you all for being out here on a Friday night, so let's get into it. So like Professor McDonald said, I pursued my master's thesis at CSU Fullerton in marine biology. So I always like to start with a little bit of what a marine biologist looks like, because I feel like the perspective of what a marine biologist is, it's a lot of like open ocean scuba diving and I'm swimming with the turtles and all the things. But most of the time it was like this. I was out in the field and had my tools, had my things, but my feet were on land at all times. So I was not an open ocean person. So what does it typically look like? Sometimes like that. Sometimes my posture is a little bit worse and I'm out doing my fieldwork things. And then people will be like, oh my gosh, you go to the beach for work? That must be so relaxing, so fun. And it is nice, but you're out there rain or shine. Sometimes I'm wearing full-on layers and it's full-on raining. Other times I think it's a peaceful day, and then other times not so much. So this is really what a marine biologist looks like on my day-to-day. Because sometimes when you wave at the ocean, it waves back at you. So this is what my day-to-day field work would look like sometimes. So no open ocean, and this is what I get for trying to avoid the water. It comes back right at me, so got me there. The reason why I start off talking about this idea of like what a marine biologist looks like is that there's a lot of subfields within emphasis, concentrations. So when you think about marine biology, it's not just one niche you can focus on. There's a lot of different areas for everyone. If you're passionate about this type of topic, there's something for everyone, or even for someone like me that prefers to be on land. So the subfield that we're going to focus on is the intertidal zone. So again, bad posture, similar type of look we have over here. So the ecosystem that I focused in was the intertidal zone. And this is where the land meets the sea. And it's a very unique environment because this is a space where you will see periods of high tide and periods of low tide. So right now, I'm doing my work during low tide because as you can see in this photo, we see a lot of land, a lot of open space. The water is far away from me this time, which is nice. And that's when I can do my work. I can clock in and go to the field and do my thing out and about and collect my data. There is a whole ecosystem of organisms that are here, algae, invertebrates, little crabs, little anemones, all the things that you typically see when you go to the beach, when you just take a moment to look down. Then I clock out during high tide. During high tide, the water is going to come in and cover all this land that you see. So you're going to hear that language a lot throughout this talk where I'll say, oh, this is during high tide or this is during low tide. So I wanted to define that really quickly so that you understand that low tide is when everything is exposed, all the living things are there that you can see, and then high tide is when it's all covered. Now, what am I actually looking at in this photo? So my study organism was mussels, and mussels are really important to this intertidal community. A lot of what I did was at the community level. So mussels have a very important role in the intertidal zone because they are there to structure and modify the habitat, and that ends up being a space where other smaller organisms can reside and live there. So in this sketch on the left, you see we have our mussel bed. This is what we call our community of mussels. We call it a bed. And we have little anemones in there. We have different types of algae, little crabs, even little baby sea stars, even like little brittle stars. Those are teeny tiny little guys. And then I have a picture on the right to show it in a different perspective. So some research has shown in the past that there can be about up to 300 different types of species of organisms in mussel beds like this. Whenever we think about environmentalism, we think about saving the planet and taking care of our Earth. The poster child really for this is the rainforest. We think about that type of environment because you see these massive trees and these massive living things that take over that space. And if you knock down those trees, you knock down everything that lives within that space. So mussel beds play a similar type of role, because if you were to wipe out the mussels, then you're wiping out all the things that live within that space. So they are also their own type of little rainforest, just on a smaller scale. So how do mussels fulfill this role? So let's go to the next slide. Okay, so mussels make byssal threads, and this is essentially their anchor line to the rock. This is their lifeline to the surface, because the intertidal zone is not forgiving. Waves come in, waves go out. There's a lot of harsh action that happens in that space. So they have to have some type of security to the land. So they make byssal threads. And I'm going to play you a video of a time lapse. So you'll see that we have a mussel here in the lab on a tile, and it actually has a foot that it'll stick out. And you'll see some subtle movements of the foot, but not dramatically, but you'll see the foot retract when it's done making a byssal thread. So what's actually happening right now is that the mussel will stick out its foot, and it's going to shape it like a trough and then pour down this liquid polymer protein. And then once that's done and the protein hardens, then the mussel will retract its foot like you see in that video, and that means the byssal thread is ready, it's done. That lifeline, that anchor line to the surface is good. So I like to think of it as like a cement truck pouring down cement, because you're pouring down this liquid, it'll eventually harden, and then you have this nice little secure line. A mussel will continue to do this process. They do it one thread at a time. They can't make multiple threads at once. And they'll probably do this to reach about 50 to 100 threads. And then they'll try to maintain that for the course of their life. And a little fun fact, some mussels can live up to about 40-something years, which is a pretty long time. And you can tell the age of a mussel similar to like rings on a tree. So if you see like little ribbed lines on a mussel, you can count those lines and you can kind of determine its age roughly. So another thing that I like to point out is that mussels like to stick with their friends. So if I have this little clip here to show 4 mussels attached to each other by byssal threads. And this is just a little side story because a lot of what I did was working with these organisms in the lab. And I had to kind of do a lot of original design and kind of mess around, lots of trials and errors throughout this journey. So I had mussels individually laid out in tanks, and I said, okay, I'm just gonna space them out. I'm gonna put them on their little tiles, just like you see in that picture. And I said, if I leave it like that, they'll just put down the byssal threads, and when I come back after a few days, they should all be on their tiles, and they should be stuck to them, done. I come back, and they're all stuck to each other. Those 4 mussels that I show you here, they were completely separated in the tanks. So what I learned very quickly, I was like, okay, why did this happen? And I guess what I learned was mussels aren't truly sessile, because we think of sessile as an organism in their adult stage. They're stuck on the surface, they're not moving, they're not going anywhere else, so they're done. But mussels are actually sedentary, so they move a little bit and they can scoot over with their muscular foot, and they can release chemical cues in the water to let them know, like, I'm by myself and I need my friends. So they have attachment issues emotionally. So this was my first obstacle of my thesis, because I was like, how do I keep them in one place? So I rubber banded them to the tile, wrapped them like a little present. So we're gonna go in here. So now we know a little bit about mussels, and we know a little bit about the intertidal zone. So mussels are occupying this space, and they're gonna experience periods of high tide and low tide. And we see right here our little display. This is where my artistic, creativity comes in. I love the use of like GIFs and things. So you'll see that throughout this presentation. And we have high tide. So this is like my little thing of like showing you, this is what high tide looks like. You have your mussels, they're with their friends, they're attached, and they're probably filter feeding and laying down threads. And another little side thing I forgot to mention is that the species that I worked with was the California mussel. And despite its name, it has a very large geographic range. So you can find that species of mussel anywhere from Alaska all the way down to Baja. So it dominates the coast completely. And they can make threads during low tide. They're the only species that I know of right now that can make byssal threads when they're exposed to sunlight and air. Other mussels have to be in this situation. They have to be submerged in water in order to make those threads. So when we transition to low tide, we're like, okay, we're exposed to air, we're exposed to some sun. So what's gonna happen? It's gonna get a little hot for them, okay? So the body temperature of a mussel during low tide is a really determining factor of their survival in the intertidal zone. So that's because they're ectotherms. So that means that their body temperature is dependent on their environment. We go out, we're endotherms. We go outside, our internal temperature stays relatively the same. We get a little hot, we get a little sweaty, maybe a little tan, hopefully for the summer. But with mussels, their body temperature will shift dramatically. So the difference in the temperature could be like 15-degree differences. So it's quite stressful during this time. And that can include a lot of determining factors. This is like solar radiation plays a factor, wind speed, surface temperature. So this is where my area of interest was. I wanted to know more about what's happening during this period of low tide. So this leads into part 1 of this talk, now that we've established a little bit of background of the environment we're in, the species we're working with, and why they're important. So I wanted to focus on the stress that's happening, because stress can be caused by environmental conditions like temperature. And temperatures outside of an animal's normal range is going to cause that stress. So then we start brainstorming, we start thinking, okay, if a mussel is faced with stresses that go beyond its ability to secure itself to the rock, so that anchor line, if it's not strong enough, then most likely they're gonna be more likely than not be ripped off the rocky shore. So again, we come back to this analogy with like a rainforest. You wipe out the mussels, you wipe out everything that lives within that space. So my goal was to understand, okay, well, what affects mussel attachment during that period of low tide where they're experiencing all this stress? So I wanted to address that and look at their attachment specifically and see how much damage can they really take before they're left hanging by a thread. And I love some puns, so I made that the title of my thesis. Little did I know, I was going to encounter a lot of obstacles that also left me hanging by a thread as well. So we'll get into that as well, okay? So what's the objective here? I'm going to walk you through the lab experiment. So we got to think of what is our goal? What are we trying to investigate? I wanted to answer this question of how does low tide temperature and exposure affect mussel attachment? But then I have to narrow down, okay, but what part of their mussel attachment? There's so many things to choose from, but that's the great thing about science is you can pick whatever you want. Just don't pick 10 things, because then you'll end up writing a dissertation and not a thesis. So I looked at 3 components. So I looked at the number of Bissell threads per day. So how many are they putting down? And are they going to conserve energy? Are they going to not put too much time into making those Bissell threads? Are they going to, in a desperate attempt, try to hold on to the rock, put out more threads? And then I looked at single Bissell thread strength. To do this, I looked at the thickness of the threads. Hello? It's fine. I looked at the single Bissell thread strength, so looked at thread thickness, and this is an image of a Bissell thread that is under a microscope, and I darkened the image just for presentation purposes, and you'll see that every thread also has that like adhesive sticky pad at the end. There's like tons and tons of papers that just look at that part of the thread, the little sticky pad at the very end. Then I also wanted to look at whole individual attachment strength. So how much force is it going to take to rip off these individuals from the surface? Now I really enjoy this part of the process because then you can get into the design, you can get into, okay, how am I going to address these questions? And again, I was someone that really enjoyed marine biology and I really liked the idea of it, but I also didn't want to be in the field 24/7. About 90% of what I did really was in the lab. So I was really excited about this because I was taking this original design that no one had done before, and I was like, okay, and I get to implement it in the lab because I didn't want to be in the field all the time being crushed by waves. I don't even like to camp. I like to glamp. I like to, but if I'm going to pack something to go somewhere, I'm booking a hotel. That's what I'm doing, right? I had some reassuring head nods for that as well. Now, again, coming with this creativity of, okay, I need to think of what treatments, what scenarios am I going to put these mussels through. I will need you guys to follow with me of understanding the language of what I put them through. So we're going to break it down bit by bit. So the first thing we have to understand is exposure and the different types of exposures that mussels go through. So we're looking at tidal predictions right now. The same way that you check the weather, you got to check the tides. If I'm going to go out and go into the field, I need to know what the tides are looking like to see how much time I actually have outside before the waves come get me. So not every low tide and high tide is the same. So you're going to see like these hills and these valleys. The very tops of the hills are the high tides. Water is covering the land. You're not going to see any organisms. And then the bottom of those hills are the low tides. So not every low tide is the same, not every high tide is the same. The distance the water will move out is not always the same. So our first exposure treatment I wanted to apply was a single exposure, and you'll notice this trend of having like a low risk day versus a high risk day. So we have single exposure where you see a low tide where mussels would actually be exposed on the first day. And I circled that in red. I'm like, okay, this day, mussels would definitely be uncovered, exposed to a low tide, but then the low tides aren't as dramatic the next 4 days, so they're probably not gonna be exposed, and they're gonna remain underwater for the next 4. So single exposure, 1 day exposed, 4 days submerged. Then we have a multiple exposure treatment. These tides are really nice. You could have yourself a nice 8-hour workday out in the field if your tides look like this. So this is a more high-risk day because the tide is low enough, the water is far out enough to where the mussels are exposed, but it looks like they're exposed 5 days in a row. So that's a little more of a stressful week for them, right? So now, let's get into temperature treatments because we're going to combine exposure and temperature when it comes to the actual lab experiment. So first, again, you're going to see this low risk situation versus a more high risk situation. We have an ambient low tide where I have all my mussels in tanks. They're attached to their tiles, thankfully, not attached to each other. And then if I have a low tide, pull them out of the tanks, and then they're just gonna sit, not be manipulated at all. I'm not gonna manipulate the temperature in the room, so they're just experiencing normal conditions, quote unquote. Now, how are we going to do this high heat simulation? Because again, this whole process is to emulate something that could happen out in the field. So this is where the creativity comes in. So I have this like makeshift heating grid that I put together, and it's just these portable hand warmers, the ones that you can charge them and put them in your pocket on a cold day, keep your hands warm. And I was like, oh, I could use this because I can control the settings, I can control how hot I can make it. I didn't want to do a hot plate situation because then I'm just heating up the mussel from underneath. I wanted to heat up the entire body of the mussel. So when you look in the grid, you'll see that a mussel's on its tile, and
I'm not sure I understand.
no. You'll see that there's 4 heaters surrounding the mussel. And is it actually warming it up? So I had borrowed a thermal image camera to show that the heat actually moves around the individual mussel. So I was like, okay, great, it's getting hot, but I'm not cooking them. Because the first time I did this, I definitely baked them, so it's fine.
Add marinara?
Add marinara? I don't know about that. I also like to be very organized. I see a lot of my current and past students in here, and they know I like to have things on a nice schedule, keep things nice and clean. So I had to do the same thing with a lab experiment. So here's a tide prediction, and I said, okay, I gotta run my experiments the same way I run my entire life. Okay, gotta have a strict schedule. And then we have our experiment running, a low tide would begin, and then there's a group of mussels that get heated in the middle of that low tide, and then the low tide would continue, and then they would go back in their tanks. So I basically was trying to simulate this like 5-hour low tide with a 2-hour heat pulse in the middle. So now we got our schedule, we understand exposure, we understand temperature. So now we're gonna walk through the 4 different scenarios I applied in the lab, and we'll go through them 2 by 2. So again, back with the little moving waves, all right? So we have a single exposure ambient low tide treatment. We also have a single exposure high heat low tide treatment. So we have our mussels on tiles, they've made threads, they're submerged in water. Then I have the low tide begin on day 1, so the water goes away. Some of the mussels are gonna get heated in that makeshift heating grid, and then some of them are not. And then the heating would stop, the low tide would continue, and then they're gonna go back in their tanks and they're gonna sit there on days 2 through 5. So we're simulating that single exposure ambient low tide and single exposure high heat low tide. Our next 2 scenarios are multiple exposure ambient low tide and then multiple exposure high heat low tide. So every day for 5 days, we have our low tide begin, a group of the mussels gets heated, the other group does not. The low tide will continue after the heating, and then they go back in their tanks. And that will happen every day for 5 days. So now we have our 4 scenarios that we put our mussels through. So let's talk about what I found and go over some major results. And I won't dive too deep into it because we have more things to discuss of all the trials and tribulations. So let's walk through our different components of strength that I looked at. Our number of Bissell threads, okay? So how many Bissell threads they're making per day. It turns out it didn't really matter what scenario you were putting them through. It wasn't enough for them to expend extra energy on pumping out those Bissell threads. They made a set amount, and there wasn't really any significant differences between those treatments at all. However, the ones that were interesting were single Bissell thread strength, so thread thickness, and the one scenario that stuck out the most was that multiple exposure, high heat, low tide. And it looked like by the end of that experiment, those threads were a lot thinner compared to any other situation. So it makes sense that thicker threads would save the mussel beds in some way. Similarly, with whole individual attachment strength, so how much force is it going to take to remove those mussels from the rock? We see a similar result to single Bissell thread strength, and we see that mussels exposed to multiple high heat low tides, it took less force to remove those from the rock. So they have in turn become a little bit weaker. So what can this tell us about the community overall? So just to preface, the heat treatment that I implemented was a 2-hour heat pulse in the middle of a 5-hour low tide. And that's really a conservative approach in the grand scheme of things, because mussels are frequently exposed to longer periods of high temperature during low tide. However, even this conservative 2-hour heat pulse, we saw some effects on their strength. So when we look at the community level, these repeated high temperature low tides and these events that could occur, that can affect the community beyond the impact of an individual organism. And we will have events like that more and more as time goes on. And we'll have things like heat waves, because some heat waves can last up to 5 days, and they can be a lot hotter than what I had implemented in the lab. The last mussel mortality event that I had witnessed during grad school was actually in Bodega Bay, and the researchers, when they talked about this event, they said, yeah, like we went out into the field and we saw all these mussels and they just looked like they were straight up cooked. And some of them were gaped completely open and there was nothing left inside the shell, completely gone. Because then also, if that's exposed, that's a free meal for a seagull right there. They don't gotta crack open the mussel shell at all. So then they start going for it as well. So the data supported the conclusion that mussels exposed to multiple consecutive low tides with high temperatures had weakened their attachment. So if we think about it, this is more likely to occur, this scenario, with the context of rising temperatures predicted with a changing climate. So our rocky shore that we have, our nice community with all these other associated organisms start to look a little bit more bare. So then our community is a little bit more damaged. So now I'm gonna transition into all the things that went wrong, okay? So I call this part no calm, just storm. There was no warning. I was just, hit with all the dramatics, okay? What you're looking at right now is a temperature data logger called a RoboMussel. This is a $130 piece of equipment, and it's completely chipped on the top. So the black part of it is just chipped. Guess what? I found tools in the intertidal that probably showed that there was some interference in that space. The green part is just epoxy, so it's just the RoboMussel, how you attach it to the shore. And we have some barnacles that settled on there, that's fine. But the chipped part was kind of alarming. I said, okay, I usually don't have this much interference where people are messing with my stuff. And then we're like, okay, we should leave tags. So we started to leave laminated tags on our equipment and say property of CSU Fullerton, and please do not interfere with the project that's going. The tag was gone as well at some point. So I was attending Fullerton between 2018 and 2021. So toward the end of my master's, I experienced COVID, and guess what? My place of work was shut down. I worked at the beach, remember? So I couldn't go to the beach. And I wasn't quite done collecting my data. So I was like, how am I gonna do this? And I tried to push really hard on this fact that, well, I worked really hard to have this collecting permit so I can still be out there and do my job. I will be safe, I will socially distance, I will do the things correctly. And I was just told, okay, well, schools are shut down, so you gotta leave your apartment, you gotta move from Fullerton back to Turlock, you have to just finish the rest of your stuff from home. And I was just like, how do I do that? You should work from home. How am I gonna do that? So it was kind of a waiting game. I said, okay, how am I gonna go back here? This is impossible. So once the beaches had reopened, I was like, hallelujah, I get to go back, finish and still graduate on time. I know that my students have felt this way before where they're like, I need to graduate yesterday. So I understand the urgency to feel like I need to be done. I said, you know what, I'm gonna replicate my lab experiment in the field. I know I can do it, I have a plan, I have a vision in mind. And then this was what was happening. So a lot of tampering with the equipment. I said, there's no way this is still happening. So this cage that I have set up, completely cut through. That is not done by a hermit crab, okay? I have tiles that I was putting out completely smashed. I've found hammers as well in the intertidal. People just take their tools and then leave them there for some reason. And then the tile on the bottom has like scraped like words on there, like putting 2020 something, I don't know. And I was like, great, now I have like a date stamp of like when this happened to me. So I really like to actually show the things that went wrong, because I feel like when you navigate science and you learn about the things that happen, all these discoveries, all the cool things, you're reading a research paper, you're reading a news article about something, you're reading the perfect version of that research, of that project, whatever it may be. You don't see this, you don't see the trials that come with research, and I think it's really important when we're doing talks like these to talk about the things that can go wrong and to persist, because you'll get there, you'll get to the end. These two articles were put out by the LA Times, and it was about collections that were happening at the intertidal. People were tide pooling a lot more, however, they were collecting in so much excess, and these are both about my field site in particular. So I was like, oh, great. So it's not, this is just me, this is happening too. So the first article was about over-collections. Crowds removing sea creatures from San Pedro tide pools put delicate ecosystem at risk. The mussel beds in this space were not these multi-layer stacked beds. They were very much single-layer beds, where if you were to remove one mussel, you could see the rock. It wasn't that you remove a mussel and there's another mussel underneath it. So it was already a very fragile ecosystem to begin with. And for some reason, when the beaches closed and then reopened, it was like, oh my gosh, we get to go outside again. And then it just became this like hobby for people to go out. And then the second article came out, and it said 45 people charged with poaching marine life from San Pedro tide pools. 45 people was a lot in one weekend, because the previous 2 years, the amount of poaching citations was less than 10. So 45 in a weekend was crazy to imagine. These are some photos from the article, and look, my poor mussels, oh my gosh, I needed those. A lot of the times when they were being confiscated from people, they were just kind of dumping them back in the beach. So it was a waste, period. One person had collected 175 pounds of mussels in one day. I know, insane. The legal limit to collect mussels at a non-protected site, and this was not protected, because there are sites where you are totally allowed to go and collect and take from the beach, and that is okay, but there's a legal limit. So the legal limit at my sites was 10 pounds per day per person. So you could take like a family of 4 and get 40 pounds of mussels and leave, and then come back the next day if you wanted to. But 175 pounds in one day, that was beyond the amount of pounds that I could collect for my research that I got approved for the year. So that just kind of puts it in perspective of how much over-collecting there was actually happening in this space. And because I was like, okay, I don't know what to do, I'm just gonna start taking pictures, because science always starts with an observation. It takes one observation to kickstart something new. I was like, okay, let's see how many people are collecting. I was like, okay, I got 1, I got 2, I got 3. This guy with the orange bag was using a gardening shovel and just like scraping the rocks and getting as many mussels as he can. Then I looked a little bit more, there's another individual hiding. There's another individual here. And then I said, you know, I'm gonna walk all the way to the other end of the site, and there was a bunch of people over there collecting as well. And I was just like, I don't know what to do at this point, because I had half of my thesis work done. I needed the other half somehow, some way. This is what my site looked like pre-COVID. As you can see, this is a great situation. I got a lot of mussels here. Not a lot of bare rock, right? So post-COVID, this is what my site started to look like. So you can see not a lot of mussels covering the rock, mostly barnacles, so the yellow organisms that you see. That's what you see. And then some seagulls, probably scavenging a little bit. So I really felt like I was in the trenches at this point. So me and my roommate slash lab mate were like, okay, we need to go out to the field and just see what we can collect for your experiments. Let's just go. And it just became like chaotic. So we would just record.
Hello?
This is the face of someone that was just like, I don't know what to do. So I really felt like, it's fine. Everything is fine. I'll figure it out. I will graduate somehow, some way. Something else I wanted to point out was that I did get the opportunity to work for a monitoring program called MARINE. So it stands for Multi-Agency Rocky Intertidal Network. And we monitored sites that have been collecting data for over 20 years. Part of what we would do is take pictures of specific areas in the beach. So if this is the spot I need to take a picture of, I click, I take a picture, I have to come back next monitoring session, take another picture at that same spot. So over 20 years of taking a picture at the same spot, you can put together a nice little timeline of how things are looking over the years. So this bench, and I think you can kind of see, the side of the bench is full of mussels. And this is pre-COVID, okay? I'm gonna show you the same rock. This is the same area. And that's what it looked like after COVID. So completely cleaned off. Another thing I wanted to point out is in that red circle, can't really see it, but there's a lot of little baby mussels. So new populations were trying to begin, and there's not much left that actually made it through after COVID because of the collections. So I was like, okay, you know what? I came up with a new approach to the thesis, and I said, okay, you know what? I have size data. I have mussel shell length size, because every time you start a research project, you need to have some baseline data. If I was going to do work at a site, I wanted to know what did it even look like from the get-go. So I had been collecting this mussel shell size population data pre-COVID through COVID. So I was like, oh, I can compare before, what did the population look like versus after COVID. So that became the second chapter of my thesis, COVID-19 related impacts on mussel populations in Southern California. So you can see we have just some size data. So the x-axis, we have two sites that we're looking at, White Point and Point Furman. Our x-axis is mussel shell length. And then our y is the number of individuals in specific sites. You can see we have a nice distribution. We have some small individuals that are 10 millimeters in length, all the way up to, we have individuals that hit about 80 millimeters in length. So some big boys in there. But then after the pandemic, we see a shift in that. So you can see that a lot more on the smaller size class end. So there was nothing beyond 60 millimeters in length. We were not seeing anything that was very large. So this is evidence now of showing people are targeting larger mussels. So that's going to have a greater impact on that community, because then those gaps that could form are going to be a lot bigger. Another thing just on the side that I thought was concerning, I started taking pictures of all these dead mussel shells, and I was like, what is happening? And I kept hearing through the grapevine that every time mussels were confiscated from someone, they would just be dumped. They would just be dumped. There was no reattachment process. There was nothing that people were really doing to kind of fix that problem. And I said, okay, well, I can measure these and see if people are actually collecting larger individuals. But just to show you how extensive this was, this little piece where the water's flowing, that's all dead mussel shells in there. And then here's another view of all that after COVID. This was not present before. So there was just a lot of differences. It was night and day going out into the field. So just to show you what those size classes look like. So this is dead mussel shell length at White Point, just one site, not a ton of data, but just to give you a perspective that these were larger individuals, all the way up to about 100 millimeters in length. So what does this tell us what was our major result as far as my thesis? So again, this accidental chapter actually became my more like favorite chapter, because I was like, wow, this is so like relevant and it's so present. So this study really served as an example of the importance of baseline observational data. And that's how science starts. When you think about your scientific method, it's like, what do you start with? Okay, I gotta observe something, ask the question, and then move on from there. So we can use that baseline data to evaluate unexpected events like this. The major result was that the probability of encountering mussels in a larger class size was going to be significantly lower than encountering a mussel in a smaller class size post-pandemic compared to pre-pandemic. So this can have, oh no, it's so sad. Well, I'll just leave that on there. So collection activities, specifically now we're talking about selective size collecting. There was bias in how we were picking these mussels. I didn't want the small ones, I want the big ones because I want to eat those ones, okay? So what's going to happen if we combine disturbances on top of this event? Then that's going to add an additional stress that we're putting on. So we have the selective size collecting. We could have natural disturbances like waves. So if we have an event like what happened with White Point, and then immediately after we get some harsh wave action, there's a lot more damage that's going to happen to this community. We can stack another human disturbance on top of this. Southern California is known for having a lot of trampling in their intertidal zone because, you know, people, it's like heavy tourist areas. So people are walking along mussel beds all the time already. So we stack natural disturbances, we stack human disturbances on top of selective size collecting. So we're going to end up with less mussels covering the shores. So is this type of human impact still relevant? And I wanted to give a shout out to someone because, you know, what I was documenting was like this peak COVID time, and people can be like, well, no, you know, well, we're not like in a peak time of COVID. Like that's probably not relevant anymore. But this is one of my friends from CSU Fullerton. She's going to graduate this year, and she looks at a different organism. She looks at an algae called the golden rockweed, so Sylvicia compressa, and this is another important organism in the intertidal zone. And it provides habitat, provides shelter to other small guys, so very similar to the role that a mussel would fill. So she specifically looks at human impact. She looks at trampling, because again, these are still heavily visited sites. People are walking along everywhere. So whether it was me between 2018 to 2021, whether it's Megan here, who's in 2024 studying this topic, human impact still remains extremely relevant because understanding that impact on intertidal species really provides insight to broader ecosystem health. So shout out to Megan, love her. So part 3, I want to end this on a positive note, okay? So we've been talking about like all the trials, okay? We want to talk about some things that were good that came out of this. Obviously I graduated, which was great. You know, I'm here now. Yay. So I want to talk about some silver linings. So this photo that you see right here is the San Juan Island. You can see not many people are walking around here. It's only 3 people. It's actually me, my advisor, and my lab mate doing work out here. But there were tourists that would come on this island in the San Juan. The San Juan is it's a very touristy place. So a lot of people come, a lot of people would visit this site. But I had population data from the San Juan as well. So I wanted to show a little bit of that, and I took this data during COVID. So you can see the size classes. Check out these guys, huge. All the way from 10 millimeters to 150 millimeters in length. These mussels were like as big as my head, okay? If I wanted to like scrape out the inside, I could make a cute little clutch. It'd be a nice little fashion statement. I know it's not as large of a sample size because I was only in the San Juan for so long, but it gives you an idea that even during COVID, this wasn't happening at every beach, at every nook and cranny of the intertidal zone. So we have June 2020, we have July 2020. Again, we got big mussels here. They're doing great. We love that. So these are 2 sites that I worked at in the San Juan. So it was nice that this type of human interference, it wasn't happening everywhere. So these guys were doing pretty good even during the pandemic. I always like to touch on a little bit of like technology in marine biology and advances that have occurred. So I showed you a picture of my damaged robo-mussel, and I want to talk about that a little bit because that was something that was just kind of starting out when I was in grad school. There was a lot of prototypes out of this type of tech. So this is developed by Northeastern University by a scientist named Brian Helmuth. He actually was the one where I would read about, and he's really the one that coined this term about like mussel beds being like a rainforest. And I just loved that analogy so much. So these data loggers, they have the same shape, they have the same size and color of an actual mussel. So they're mimicking that biology. And they have these built-in sensors, as you can see in that sketch, and that helps track temperatures within the mussel beds. So that can help us understand how temperature changes affect mussel populations. Because you can study this data that's collected by these little robo-mussels, and the researchers can gain insights into the impacts of climate change and at the community level. Because again, these are placed within the mussel beds. And since I've graduated, there is actually a website now where people can look up where these loggers are deployed, and they're deployed all like worldwide. So you can select a region and look at the data and see the temperature fluctuations there. So how do you get that data? So I have a little clip to show you. Let's see if that'll play. So this is what a robo-mussel looks like, and it actually is a Bluetooth technology moment. So you can take that, put the little sensor on. You can set it to record every 10 minutes if you wanted to. You can set it, and then you can place it in the intertidal, and then you can collect that data again, do the Bluetooth little tap, download it, email it to yourself, and you just have a bunch of temperature data, which was really cool. So I really wanted to try these out and test these out, but it's like knowing my luck, like of course something kind of went wrong with it. So I'm going to tell you that story. So I got to correspond with one of the scientists that made robo-mussels. His name was Fernando Lima, and I was like, oh, I get to work with this like scientist that I'm reading about in papers. I was like a little intimidated. I was like, oh my gosh, what am I going to do? Can I buy like 12 robo-mussels for my thesis project? And he was the most humble, sweetest guy. He was based in Portugal, and we corresponded a little bit about what I needed for my project. I was able to get a batch of like 12 robo-mussels from him. And I was like, okay, whatever I get from this, I'm gonna, let you know what data I get from it, because I think that would be really cool. I set them out in different places. I go back and check on them, and none of them are working, which is great. So I was like, how am I going to approach this big scientist that I was like reading about and tell him, okay, all your robo-mussels that you gave me are broken. I broke them, I'm sorry. He's like, just send them back and we'll see what went wrong. And I was like, okay, like crying on the inside. I was like, I just broke this equipment. And he emails me back, he goes, oh, it looks like there was a flaw in the design and then seawater like seeped into the data logger, which destroyed the battery. And I was like, oh, okay. And he goes, here's our new design, and this is our new prototype. We're gonna send you a new batch of robo-mussels. And I was like, wow, I helped, you're welcome. Thank you. So it just goes to show at any level of science, like there's things that are gonna go right, there's things that are gonna go wrong, and that's okay. Now I wanted to show you a little bit of some of the long-term data that Marine has collected. Now again, some of these sites have been monitored for 20 years. So I know this looks like a lot of data points, but we're gonna focus on pre and post-COVID. So this is looking at mussel cover at the site that dealt with all that human interference, like where I worked at. So percent cover is if I were to take like a frame and I take it out to the intertidal and I place it down, and if every point in that frame has a mussel in it, that's 100% cover. If only half of that square is filled with mussels, then that's only 50% cover. So we want 100%. You can see there's a gap in the data where COVID happens because we couldn't do our work. But it looks like that we're on an uptrend, which is really nice to see that the mussels are getting back. So maybe we're having mussels that they're better withstanding this human impact. Maybe they're withstanding thermal stress a little bit better in the hopes that they'll have greater chances of survival. And then over time, maybe they'll be able to be a little more resilient. So this is really nice to see. Now, is that going to affect other species as well? So this is still percent cover, and there's 3 lines here, and it even tracks a line for rock. We don't want rock to be at 100% cover. If I take a frame out in the intertidal, I want to see a bunch of organisms, right? So we want that to go down. So pre-COVID and to after COVID, we see that the percent cover of rock is going down, which is great. And then we have 2 species of barnacles that reside in mussel beds, and that's on an uptrend too after COVID. So it looks like we're on an uptrend, which is really nice. See, silver linings, not everything's going wrong. I will leave you with just a few closing thoughts, and that is, the first one is, I'm not here to tell you you can never collect and eat another mussel again. Okay, I understand it's something that people like to do. But I want people to do this safely, because the timing of when I saw these people collecting these mussels was not a safe time to be eating them at all. Mussels are the most dangerous when it comes to biotoxins. They accumulate the toxin way quicker than oysters, way quicker than clams. And health officials have time and time again have said you cannot distinguish a toxic mussel versus a non-toxic mussel. And you cannot cook away that toxin because it's heat resistant. So I put up this little QR code for you guys, and it's just going to take you to the website of California's biotoxin hotline, because if you are at a site and you want to collect at a non-protected site, right, not a protected site, got to do the place where you're actually allowed to collect, you can call the hotline and say, hey, is it a safe time for me to collect this organism or that organism? And they'll tell you how recent that information was updated. Now, another thing that was alarming was seeing all those dead mussel shells, and I showed that with purpose of talking about participating in coastal cleanups. So I have the link for our California Coastal Cleanup Day, and this is the highlight of California's Coastal Commission's year-round Adopt a Beach program. This was adopted in 1985, and it's only gained more and more traction. And they have been getting at least 70,000 volunteers annually. They even have broken a world record of most garbage collected in one event. So that's really nice. The last thing I have is a link to Laguna Ocean Foundation, and it's a really nice page that goes over how to safely tide pool. Because again, I'm not here to tell you, I never want to see you at the beach again because you ruined my project. It wasn't you. This was one incident. This is science. This is what happens, okay? But you have to practice safe tide pooling practices, okay? We have this picture of these little kids. They have little buckets. They're at a non-protected site. They can collect some things. So I always say when you're at the intertidal, be cautious in these areas. Respect the wildlife out there. If you're going to collect within the legal limit, not 175 pounds of mussels. That's a lot. So stick to your marked paths. If a living thing, if an octopus looks dangerous, don't approach the octopus. I've had a colleague stick their keys in front of an octopus because they like shiny things. So they'll snatch them from you. So. And it did, so I was like, we shouldn't probably do that. Wear the right shoes, right? I've seen people barefoot out in the intertidal, don't do that. And have a buddy check the weather, check the tides, right? Check how far out is that water gonna be, how much time do you have out there? And then if you're allowed to collect or even touch an anemone, use the 2-finger touch rule. You can give it a little anemone hug, and that's fine. But be safe while being out there. I do want to put some acknowledgments on the board and some list of funding materials that help with this project, and that is my talk. Thank you. Okay, so if people have